symposium, it falls to me to introduce these cells, the oligodendrocyte progenitor cells, or OPCs. Now, it'll advance. Okay, so during mouse embryonic brain development and also during human gestation, it's the radioglial stem cells that give rise to these OPCs. Now, once born, they proliferate and migrate to populate the central nervous system, and after birth give rise to the myelinating oligodendrocytes. Now, we often depict OPCs as quite simple cells, with often only drawing them with a couple of processes. But in reality, they're actually highly complex cells, as shown here by this one that's been transgenically labeled with a membrane-tagged fluorescent protein. And it's sitting next to a myelinating oligodendrocyte. So OPCs, though, they're not existing in isolation either. They're actually forming a mosaic of cells that span the central nervous system. And this is perhaps best depicted in this coronal brain section here, where each of the little black dots is an OPC, and you can see that they're quite evenly spaced throughout the brain. So by performing cranial window surgery and live 2-photon imaging, members of Dwight Burkle's lab were able to visualize the OPCs and monitor their behavior in real time. And what they found is that, like, an individual cell body of the OPC might not be moving very much, but each cell is sending out processes and retracting processes quite actively. And when processes from two adjacent OPCs, shown here as a green cell and a pink cell, come into contact, those processes will touch for a short amount of time before they're then withdrawn. And this process of presumably contact-mediated signaling that's facilitating this withdrawal allows the OPCs to maintain discrete domains within the brain. And this process is also really important for restricting oligodendrocyte progenitor cell proliferation. So my team was wondering, what are the key signals that might be responsible for regulating this function in OPCs? We looked at some RNA sequencing data that was available from the Barras lab, and we found that protocadherin 15, a member of the cadherin superfamily of calcium-dependent adhesion molecules, was actually highly expressed by OPCs, but also quite selectively expressed by OPCs. So we thought maybe this is actually a key signaling molecule in this contact-mediated signaling. So what is protocadherin 15 and what does it look like? Well, it's shown here as a dimer. So this is a cell membrane, and it's got a short cytoplasmic domain, a single transmembrane domain, and then these 11 extracellular cadherin repeats. And it's best known for its function actually in the ear, where it's expressed by auditory hair cells. And it binds to cadherin 23, forming a really stable structure. It's called a tip link, and it joins the stereocilia and allows mechanoelectrical transduction in hair cells. So it allows us to hear. But it's also got one known role within the central nervous system, and this is actually in pathology. So it's been shown that in people that have glioma, if their glioma has a low level of protocadherin 15 expression, then this is associated with poorer survival outcomes. The flip side of that is that in a xenograft experiment where where the researchers took glioma cells and transplanted them into the mouse brain, when protocadherin 15 was overexpressed in those glioma cells, it actually reduced the tumor growth and increased the survival of the mice. So the control survival is here in red, and the overexpression survival is here in blue. And so since we know that OPCs can, like glioma cells can have OPC genes expressed as part of them, and OPCs are likely some of the cells that could be responsible for glioma development, we thought that protocadherin 15 is therefore an even more likely candidate to be regulating perhaps some of this contact-mediated signaling and ultimately OPC proliferation. To explore this directly, we turned to mouse primary OPCs, generating cultures that look like this one up here in the corner from the early postnatal mouse cortex. Now, these cells are just stained to detect protocadherin 15, but the cultures were then treated with lentiviral particles to knock down protocadherin 15. And so if we look at the Western blot data, cells that were treated with this control scrambled shRNA and stained to detect protocadherin 15, you can see quite a lot of different bands here, and this corresponds to the number of different isoforms of protocadherin 15 that are expressed by the cells. But when we gave the protocadherin 15 shRNA, we actually found that all of the different isoforms could be knocked down across the board. And this was achieved within 12 hours of giving the shRNA. So it allowed us to then say, okay, we've now got cells that have protocadherin 15 knocked down. What does that do to their proliferation? So we added EDU into the cultures, and each OPC is shown here by a GFP positive nucleus. And the EDU, so the thymidine analog, which marks the cells that have divided, is shown in red. And when we quantify the fraction of OPCs that have divided within a 12-hour period, we found that protocadherin 15 knockdown cells divided twice as quickly, or twice as many of the cells had divided. We know from the literature that mitogenic signals in OPCs, the different receptors that, like PDGF receptor alpha, that drives OPC proliferation, often does this by activating MAP kinase signaling and Erk phosphorylation. So we thought, well, maybe protocadherin 15 is normally suppressing this pathway to restrict proliferation. And so we looked at the expression of Erk1 and 2 in control and knockdown OPCs and found that the total protein levels were completely the same. But we did find that both phospho-Erk1 and phospho-Erk2 were increased in the knockdown cultures. And so our goal was then to block Erk phosphorylation and see whether or not protocadherin 15 knockdown could or could not then still drive extra proliferation. And so we gave this drug U0126, which is a selective MEK inhibitor, which blocks Erk phosphorylation. And as you can see, that's confirmed here in the cultures that were treated with the drug. Erk phosphorylation is almost abolished. And we again looked at the OPC proliferation, quantifying the proportion of these GFP nuclei that were EDU labeled. The knockdown cultures still had significantly more proliferation than the controls. But when we look at the U0126 treated cultures, the control and the protocadherin 15 knockdown cultures had the same level of proliferation. So protocadherin 15 knockdown was no longer able to push that level of proliferation up. So we believe this is the pathway by which protocadherin 15 knockdown is normally allowing the cells to divide more quickly. But as I mentioned, we were really interested in this contact-mediated signaling and OPC motility. And so we performed a series of time-lapse imaging experiments, zooming in on individual OPC processes and actually monitoring their motility over time. So like neurons, OPC processes actually elaborate these actin-rich lamellipodial-like structures, which get thrown out and pulled back in, sensory structures at the end of the process. For OPCs for this project, we're calling them veiling or veils. And so when an OPC sends out that lamellipodial-like structure and pulls it back in, I'm calling it veiling. And we measured the amount of time that the veil was extended for, like a full loop of extension and retraction in control and knockdown cultures. And we found that this occurred really quickly in the control cultures, but was really very slow in the protocadherin 15 knockdown cultures. We also then got a better camera for our time-lapse imaging microscope, and we're able to look at the fine filopodia that are actually extended from those structures that contact between adjacent OPCs. And we identified points where those filopodia touch, and then we measured the amount of time they stayed in contact for. And we found that the protocadherin 15 knockdown cultures, when the contacts were made, they were actually maintained for an unusually long period of time. And that was reflected by our quantification. So what signaling is actually downstream of protocadherin 15 that normally allows really rapid contact-mediated withdrawal? We do know that Erk phosphorylation in some contexts can regulate OPC motility. So we initially looked at what happened to these cultures if we treated them with the same drug, U0126, to prevent Erk phosphorylation. And what we found was that actually this had no effect whatsoever on the motility phenotype. So if we looked at veiling time, the knockdown OPCs, their veiling was really long, so that kinetics is slowed down a lot. And this was still the same even when Erk phosphorylation was blocked. Consistent with this, when we looked at these Filopodial contact time, again, it was increased in the knockdown cultures, but preventing Erk phosphorylation had absolutely no effect on this whatsoever, so that we know protocadherin 15 is regulating a different pathway in the processes of these cells. So we weren't really sure what was happening, so we had a little look by immunostaining these cells to detect actin, shown here in red, at the end of the OPC processes, and microtubules, shown here in green. And what we found in the knockdown cultures was that we got this accumulation of actin at the end of the OPC processes. So to find out what might be responsible for that, we carried out a qPCR on cultures, on 3 independent cultures that had been treated with shRNA for control or knockdown, or protocadherin 15 knockdown. And we found a small number of genes that had only been sort of mildly upregulated. But we also noted that a lot of them were associated with CDC42 to ARP23 signaling. So that pathway that does lead to actin polymerization. Now, this was consistent with what we'd seen from our immunostaining. So we thought we should validate this by looking at proteins relevant to this signaling pathway. We performed Western blots on our control cultures as well as our protocadherin 15 knockdown cultures, looking for proteins like CRK2 and WAVE2 that are upstream of ARP23, so they sit within this part of the pathway. And we found that these proteins were significantly upregulated following protocadherin 15 knockdown. We also looked at components of the ARP23 complex itself, so ARP2, ARPC3 and ARP3, which actually combine to form this ARP23 complex. We found that they were also upregulated in our knockdown cultures. So we thought, okay, when we lose protocadherin 15 signaling, we're getting additional actin polymerization via this pathway. Does this make sense based on the localization of protocadherin 15 and ARP23 within the cell? So we performed some higher magnification imaging, and we found that protocadherin 15 does actually extend within the processes of OPCs. And we stained for a number of proteins that are part of that ARP23 complex. And here we show ARP2 is also extended throughout the OPC process. So we wanted to find out whether this could really be responsible for what we're seeing. And so we again turned to pharmacological inhibitors, blocking CDC42, so upstream here with ML141, and blocking ARP23 with this drug CK666. And in cultures that had been treated with these drugs, we found that the amount of actin accumulating in the processes of OPCs could actually be rescued, so that the actin levels in our knockdown cultures came back to that of controls following treatment with either drug. So we were able to overcome this actin accumulation. But is it enough to rescue the motility? So to look at that, we just selected one drug here, the ML141, so the one that's further upstream. And we treated our cultures with the CDC42 inhibitor, and went on to again look at time-lapse imaging and veiling, and quantifying the amount of time taken for these veils to take to be extended and then retracted. And we found that in our protocadherin 15 knockdown cultures, which had slower veiling time, that veiling time could be rescued by the treatment with ML141 to offset the effect of protocadherin 15 knockdown. And when we looked at specifically at the Filopodia again and looked at that contact time, we found that in our protocadherin 15 knockdown cultures, again, that contact time was longer, so the contact was sustained. But when we treated with ML141, we could again rescue the phenotype. So overall, we conclude that protocadherin 15 is a negative regulator of distinct signaling pathways, one of which is the MAP kinase Erk signaling pathway, by which point it modulates OPC proliferation, suppressing OPC proliferation. And in the processes, protocadherin 15 also is a negative regulator of this CDC42 ARP2/3 signaling pathway, so that it reduces actin polymerization and allows this really rapid veiling and motility, as well as contact-mediated repulsion. So to acknowledge the work that was done by different members of the lab, the person I really most need to highlight is Alan Zen, who undertook this project as a PhD student in my lab and is now graduated, as well as collaborators of the project, Rob Gasparini, Carly Cullen, Ben Emery, and Ahmed Zubair, without whose help we could not have carried out this research. And the research, this is sort of a sample, I guess, of what's in the paper, but has been recently accepted for publication. So hopefully the full story will be out there soon. Thank you.